COVID-19 cases, deaths, and new XEC variant spark concern across the UK. England's health officials report a rise in hospitalizations due to COVID-19, with deaths increasing by over 25%. Scientists are analysing the newly detected XEC variant to assess its potential risks. Doctors have also observed a surge in respiratory infections, including colds, flu, and COVID-19, as the country grapples with widespread illness, often referred to as the lurgy. According to the UK Health Security Agency (UKHSA), COVID-19 test positivity in hospitals rose to 14.6%, up from 13.5% the previous week. Hospitalization rates have increased to 4.6 per 100,000, compared to 4.46 the week before and 3.722 weeks earlier, indicating a clear upward trend. Between October 2 to 9th, COVID-19 cases increased by 17.8% to 3,496, while deaths rose by 27.3% to 163 as of October 4. There are now 2,622 hospitalized COVID patients, 6% more than the previous week, with 68 acute respiratory incidents reported. Test positivity remains highest among those aged 85 and older, with a 23.7% positivity rate, up from the prior week. The northeast region of England currently has the highest hospitalization rate, at 8.91 per 100,000. Among those aged 85 and older, admissions have remained steady at 52.65 per 100,000. Vaccination efforts show that 8.5% of under 65s in clinical risk groups and 24.7% of people aged over 65 have received an autumn 2024 booster shot. Dr. Jamie Lopez-Benol, consultant epidemiologist at the UKHSA, urges eligible individuals to get vaccinated against COVID-19, flu, and RSV. Now is the time to protect yourself and stay strong this winter, he emphasized. The XEC variant, first detected in Germany in June, has since spread to the UK, Denmark, and the US. Current surveillance suggests that around 10% of sequenced COVID cases in the UK belong to the XEC lineage. While the variant is being closely monitored, experts, including Dr. Monica Gandhi of the University of California, indicate that it displays symptoms similar to other Omicron subvariants, such as sore throat, fever, body aches, and loss of smell or appetite. The NHS advises individuals with COVID symptoms, including a high temperature, continuous cough, or fatigue, to avoid contact with others for at least 5 days, especially with vulnerable individuals for up to 10 days after testing positive. Although self-isolation is no longer mandatory, those feeling unwell are encouraged to stay home until they recover. If children have mild symptoms but feel well enough, they can attend school or childcare, according to the NHS. Vaccines are expected to continue offering effective protection against severe illness from the XEC variant and other circulating strains. New research links shingles virus to increased risk of Alzheimer's disease. Shingles virus linked to increased dementia risk, new research suggests. Scientists are exploring the possibility that common viral infections may contribute to dementia. Cutting-edge research indicates that shingles, a virus infecting about 1 million Americans each year, could increase the risk of developing Alzheimer's disease. Researchers found that people who received a shingles vaccine were 20% less likely to develop the condition years later. This emerging area of study focuses on the connection between viruses like chickenpox, shingles, and herpes with dementia. Shingles, caused by the varicella-zoster virus, the same virus responsible for chickenpox, remains dormant in the nervous system after the initial infection. According to the Mayo Clinic, the virus can randomly reactivate as people age, triggering painful rashes and nerve inflammation. Although the cause of reactivation is unclear, it appears to be linked to a weakened immune system, as it is more common in older adults or individuals with other illnesses. The Centers for Disease Control and Prevention, CDC, reports that about 1 million people in the US develop shingles annually, while 500,000 Americans are diagnosed with Alzheimer's disease each year. Despite Alzheimer's being so prevalent, research into its causes has made limited progress. However, Dr. Pascal Geldsetzer from Stanford University offers a new perspective, examining how viral infections might contribute to the disease. Geldsetzer's study, which is awaiting peer review, analysed 300,000 health records of people born in Wales between 1925 and 1942. By comparing two groups, one born before 1933, who didn't receive shingles vaccines, and another born after 1933, who did, the researchers found that vaccinated individuals had a 20% lower risk of developing dementia within 7 years. They verified these findings by conducting similar analyses in the UK and Australia, observing the same trends. At Oxford University, a 2024 study published in Nature also supports the theory. Researchers analysed 200,000 Americans who received Shingrix, a shingles vaccine introduced in 2017. Results showed that vaccinated individuals experienced a 17% reduced risk of dementia over 6 years compared to those who received older, less effective vaccines. Paul Harrison, a professor of psychiatry at Oxford, highlighted the potential long-term benefits of vaccination. Vaccines may offer more than just short-term protection, they could have lasting health effects. While the connection between shingles and Alzheimer's is still under investigation, researchers believe the virus's behavior in the nervous system may play a role. Though the varicella-zoster virus seems inactive after chickenpox, studies from the Netherlands suggest the immune system continuously suppresses it. As immunity declines with age or illness, the virus may reactivate, possibly disrupting blood flow to the brain. Neurologists from the University of Alabama have shown that reduced blood flow over time can damage brain cells, contributing to cognitive decline. Dr. Maria Nagel, a neurovirologist from the University of Colorado, expressed optimism about the new research direction. For years, Alzheimer's research focused on a single theory. Exploring how viruses contribute to neurodegeneration opens exciting new opportunities. With more scientists investigating the viral link to dementia, Dr. Nagel is hopeful about future breakthroughs. I believe that in the next 10 years, we'll see significant progress in understanding new mechanisms and finding ways to slow the progression of Alzheimer's. Bird flu outbreak sparks food safety warning. Experts advise caution with eggs, dairy, and poultry. A former FDA food safety expert, Dr. Darin Detweiler, is advising Americans to change their grocery and cooking habits as bird flu continues to spread across dairy farms in the U.S. In California, 6 cases of bird flu have been reported among dairy farms over the past 3 weeks, with a total of 90 herds now affected. Although the H5N1 virus has not been officially linked to infections through food, an isolated case in Missouri earlier this year could not be ruled out. Dr. Detweiler warns that raw or undercooked eggs present the highest risk, recommending that people avoid dishes like soft-boiled eggs, poached eggs, or eggs Benedict. Stick to fully cooked options such as hard-boiled eggs, scrambled eggs, and well-done omelets, he said. He also advises against using egg-based condiments like mayonnaise and aioli, as well as desserts like mousse and tiramisu, which often require raw eggs. Until the outbreak is under control, it's safer to avoid foods that rely on raw or lightly cooked eggs, Detweiler adds. Salad dressings with raw eggs, such as Caesar dressing or hollandaise sauce, should also be avoided, especially when dining out. Safer alternatives include olive oil or vinaigrette. Dr. Detweiler cautions that even at steakhouses, there are potential cross-contamination risks from mixed-use kitchens that handle both poultry and beef. He encourages consumers to check restaurant safety ratings on local health department websites. When shopping for groceries, Dr. Detweiler recommends choosing pasteurized products, which kill harmful bacteria and viruses. He advises against consuming liquid egg whites, raw cookie dough, unpasteurized eggnog, and soft cheeses made with raw milk. He also suggests checking poultry packaging carefully for signs of mishandling. For those looking to reduce their risk further, Dr. Detweiler notes that going dairy-free is now easier, with many plant-based alternatives available. Given the infections among dairy workers, plant-based milks and cheeses are worth considering, he said, adding that plant-based salad dressings are another option. The bird flu outbreak in California is already causing ripple effects nationwide, as the state is a key supplier of eggs and dairy products. If California's supply decreases, other regions may rely on pricier alternatives, driving up food costs across the U.S., Detweiler explained. The outbreak may also impact the availability of turkeys for Thanksgiving and Christmas. According to the CDC, 6 human cases of bird flu were reported among dairy workers in 2024, with 9 more cases tied to poultry exposure since 2022. One person was infected without any known contact with livestock. The CDC warns that more human cases are likely in the coming months, with the virus spreading quickly among dairy herds in at least 14 states. While the current risk to the public is considered low, experts worry that the virus could evolve into a pandemic threat similar to COVID-19. New study reveals 2-phase progression of Alzheimer's disease. A groundbreaking study, funded by the National Institutes of Health, NI, and published in Nature Neuroscience, reveals that Alzheimer's disease progresses in 2 distinct phases. Researchers used advanced brain mapping techniques to analyze over 3.4 million individual brain cells, shedding new light on how the disease develops over time. 2-phase process of brain damage. In the first phase, which begins years before symptoms emerge, specific brain cells, particularly inhibitory neurons, gradually deteriorate. These neurons help regulate brain activity, and their loss may destabilize neural circuits, setting the stage for further brain damage. Surprisingly, these inhibitory neurons, rather than excitatory neurons, previously thought to be the primary victims, are among the first to be affected. The second phase occurs when Alzheimer's accelerates, marked by widespread inflammation, cell death, and the rapid buildup of plaques and tangles. This phase aligns with the onset of memory loss and cognitive decline, indicating that significant brain damage has already occurred by the time symptoms appear. Early brain changes offer hope for intervention. The study highlights that much of the early damage is silent, meaning it happens without noticeable symptoms. Plaques begin to form slowly, and the brain's immune system activates, but structural damage remains minimal. Researchers believe this early period presents an opportunity for intervention, potentially halting or slowing disease progression before severe symptoms develop. New discoveries shift Alzheimer's research focus. The study analyzed brain tissue from 84 donors at various stages of the disease, focusing on the middle temporal gyrus, an area crucial for memory, language, and vision. The findings challenge long-held views by demonstrating that the loss of inhibitory neurons, not excitatory ones, drives early brain damage. This shift in understanding could pave the way for new treatment strategies. Additional insights and genetic factors. A related NI-funded study at MIT identified a gene called relin, which may influence how susceptible certain neurons are to Alzheimer's. Star-shaped brain cells called astrocytes, thought to play a protective role, also emerged as key players in both studies. Challenges and future directions. While the study offers critical insights, it focuses primarily on the middle temporal gyrus. Future research aims to explore other brain regions to develop a more comprehensive map of Alzheimer's progression. Additionally, researchers emphasize the need for real-time detection methods, such as brain scans or blood tests, to identify the disease in its earliest stages in living patients. This research represents a major step forward in understanding the cellular changes that occur in Alzheimer's, said John Nye, director of the NIH's Brain Research through Advancing Innovative Neurotechnologies, BRAIN initiative. By identifying early changes, we can target treatments to specific stages of the disease and develop new diagnostics that offer hope for patients and caregivers worldwide. Moving forward, scientists hope to explore how genetic factors, like the APOE4 gene, which raises Alzheimer's risk, interact with these cellular changes. The ultimate goal is to identify at-risk individuals early and develop targeted interventions before symptoms emerge. Parasitic worms and viruses, a new look at the causes of severe disease. Roundworms, or nematodes, are a primary cause of parasitic infections globally, leading to painful swelling, intense abdominal pain, and even blindness. Recent research suggests that the viruses carried by these worms may contribute significantly to the severity of these illnesses. A study published in September in Nature Microbiology examined over 40 species of parasitic nematodes, focusing on a molecule known as RNA. This molecule, similar to DNA, is essential for protein synthesis and serves as the foundation for many viruses. Researchers identified 91 RNA viruses across 28 of the studied worm species, which accounts for about 70% of the roundworm species known to infect humans. Elodie Gedin, a parasitologist and microbiologist at the National Institutes of Health who was not part of the study, remarked that this research marks the beginning of an exciting new avenue in virology and pathology. Lead author Shannon Quek, a parasitologist at the Liverpool School of Tropical Medicine, LSTM, in the UK, originally researched viruses in mosquitoes and developed a method for detecting viruses in nematodes. Quek created an algorithm designed to identify viral signatures in mosquito RNA. In a moment of inspiration, he applied the same technique to the transcriptomes of parasitic nematodes, essentially the complete RNA profiles of the worms. By analyzing publicly available roundworm data, the team searched for a specific marker present in all RNA viruses, a molecule encoding a particular enzyme. This approach led to the discovery of viral genetic material embedded within the worms. Out of the more than 90 viruses identified, only 5 had been documented before, the majority were newly discovered. The research team then investigated whether these viruses were replicating within the worms. They focused on 2 species, Brugia malae, which causes limb swelling, and Onchocerca volvulus, responsible for skin diseases and blindness. Under the microscope, they observed the presence of viruses in both types of worms, specifically, the BMRV1 virus in B. malae and OVRV1 in O. volvulus. In the case of B. malae, the researchers noted an elevated immune response against BMRV1. Additionally, when they processed B. malae worms, they detected viral proteins, indicating active replication of these viruses. The crucial question remained, do these viruses influence the health of the humans infected by these nematodes? To explore this, the researchers collected serum from individuals infected with either worm and found antibodies targeting the worm-associated viruses. This suggests that the human immune system reacts to these viral pathogens, and given that these viruses have only been found in the worms, human exposure from other sources is unlikely. The next step for the researchers is to determine whether the viruses contribute to symptoms associated with these parasitic worms. For instance, O. volvulus has been linked to onchocerciasis-associated epilepsy, OAE, a condition that results in seizures. While the worm's role in these neurological symptoms is not yet understood, OVRV1 may be a potential factor. Mark Taylor, a senior author of the study, suggested that OVRV1 could be the key driver behind the disease. Rhabdoviruses, which include the rabies virus, are known to infect and harm nerve cells, making OVRV1 a strong candidate for causing OAE. Moreover, some parasitic worms are known to carry bacteria such as Wolbachia, which is also associated with diseases linked to roundworms. The scenario of a parasitic infection involves more than just the worm itself, it includes the presence of bacteria within the worm, along with any viruses, Gedin explained. All of these entities replicate and produce proteins that can influence the host's immune response. Study reveals DNA alterations from high-potency cannabis use. Research from King's College London and the University of Exeter indicates that high-potency cannabis has a significant impact on the DNA of users. The study identified unique alterations in DNA that could help understand the biological effects of cannabis consumption. This finding raises the possibility of developing tests to identify individuals who may be at risk of experiencing psychosis as a result of cannabis use. High-potency cannabis is defined as having a tetrahydronaban, THC, concentration of 10% or higher. THC, the psychoactive element in cannabis, is responsible for the euphoric effects, but at elevated levels, it can also lead to anxiety and paranoia. Since the 1990s, THC levels have been on the rise in both the UK and US, with products available in Colorado, where cannabis is legal, containing up to 90% THC. Previous research has indicated that individuals who consume strong cannabis may face a heightened risk of developing psychosis or schizophrenia, although the degree of this risk remains debated. Adolescents are particularly vulnerable as their brains are still developing during this crucial period. The researchers found that high-potency cannabis impacts DNA methylation, a process that regulates gene expression. Specifically, the study noted alterations in genes associated with energy metabolism and immune responses. Participants experiencing their first episode of psychosis displayed different DNA changes compared to those without such experiences, particularly among high-potency cannabis users. This indicates potential for DNA blood tests to identify cannabis users at increased risk for psychosis, facilitating preventive measures. Marta Di40, a professor specializing in drugs, genes, and psychosis at King's College London and the report's senior author, emphasized the urgent need to comprehend the biological effects of cannabis, especially on mental health, given the rising popularity and availability of high-potency varieties. Our research is the first to demonstrate that high-potency cannabis leaves a distinctive DNA signature linked to immune and energy production mechanisms, she said. Dr. Emma Dempster, a senior lecturer at the University of Exeter and the study's lead author, remarked, "This groundbreaking study highlights that frequent use of high-potency cannabis produces a unique molecular imprint on DNA, particularly affecting genes related to energy and immune function. Our findings shed light on how cannabis use may modify biological processes. DNA methylation serves as a crucial link between genetic predispositions and environmental influences, enabling external factors, like substance use, to alter gene activity." Thank you for joining us for the latest roundup of major health headlines from around the globe. If you found this video informative, please give us a thumbs up and subscribe to our channel. Your support helps us bring you the most current health news. Remember to stay happy and stay safe until our next video. See you soon.